So in this short video we'll see how the sequencing using the Illumina technology works. So this sequencing is called by Illumina sequencing by synthesis. And the reason for that is that, in fact, we determine the sequence of a fragment or of a piece of a fragment while we synthesize it. Okay, so imagine that you have this piece of DNA that you want to sequence. So the first step, while we are doing the library preparation, is to attach on this piece of DNA two adapters, so two oligos, let's call them A and B. So then we have the flow cell, or the whatever we call the glass, or this chip here. It's not a chip actually, but this platform in which we have attached millions, hundreds of millions of these adapters. And in fact these are in pairs, so next to each other. No, this is not correct. So we have attached all these adapters. They are not in pairs and they are not next to each other. This was my mistake. But there are hundreds of millions of that. I have to mention here that this piece of DNA that we have taken from a big chromosome via sonication, so we have make breaks on this initial DNA, and for example this one is this blue guy here. We have incorporated the adapters A and B here by ligation. So from this step to this one we go via ligation. And to break this one we have used sonication. Okay, so we have attached here these adapters and we have our flow cell, or whatever, this platform, with the adapters also attached to it very firmly. So the next step is that this molecule will be attached here to its complement part. So let's say that this red here is complementary to this red here, which means that what we will get eventually is something like that. So these two guys are complementary to each other. I use the same color, they don't mean that they are the same, they are complementary, okay? And then it will be like that. So this molecule has been attached this way. Now regarding this pair here, we should not forget that this is attached. Is attached to the platform, while this is not attached because it's just hybridization here. And then we synthesize this guy here. This we can do it. So again it will be reverse complement. Or it will be complementary. And then the next step, so first step, step number 1 is synthesis. So if we start from the beginning, we will have sonication, and ligation. Ligation is here to attach these adapters on this piece of DNA, and sonication is here. These are all for the library preparation. Then we have this platform with the oligos attached to it. And then we have the hybridization of this piece of DNA here. So step 3 is hybridization. And then step 4 is synthesis of the reverse complement. So this one. So the synthesis starts from something that's attached, while the original one is not attached. So the step 5 is wash away the original. Since this piece of DNA is not attached, it's easy to wash it. So I will just delete it. And then we have the bending of this, so that it will bend to its complement. It will go like that, it will come here and will bend here. Again, this green is complementary to this green here. So again I have to and now we have the synthesis of this one up to here. So the synthesis starts with polymerase from this point, using this as primer to start to initiate the action of polymerase. We have it already here. So we will get this one. Now let's call it molecule number 2 and this molecule number 1. Molecule number 2 is attached here, while molecule number 1 is attached here. The other ends are free. So then we denaturate it, we dehybridize them, and we will have something like that. Let me delete it again. So this, it will be green here, and it will be red here, it will be blue in the middle. So since these are attached to the to the platform, they cannot move, and it will be like that. And then this process continues, so this will bridge here. This red is complementary to this one, and this will be bridged somewhere here. Again, I have to delete these guys because they are bridged. Then synthesis again will take place, and then we will dehybridize these two guys, and from one molecule in the beginning we will go to a cluster of clones, as we said in the class. And after the formation of clusters, so this is something that we didn't say in the class. So step 6 here is synthesis of the, or better it's bridge formation and synthesis, and eventually this will lead to the formation of clusters. And now in step number 9 we wash away all molecules that are in one of the two directions, either the forward or the reverse. Let's say the reverse direction. Which means here that if we have a cluster like that, let's draw it again. So here is the platform. We have the red guys here. So let's suppose that this is a cluster. The green one here, and we have done all the previous steps. So we have this small cluster here. Then we wash away the ones that are in a certain direction, let's say the reverse one, which means that we delete this guy, for example, all the ones that are attached in green. This guy, yeah, let's better do it like that. So we delete this one, washed away, delete this one, washed away, delete this one, washed away, and this one. And we remain with these ones here that are now identical, exactly identical, in the correct direction. And now we start the synthesis of this piece of DNA here. So now in the sequencing by synthesis we do the following. We use primer that is attached, can attach here, okay, and we'll start sequencing in this direction. Now let's take an example of these molecules here just to describe in more detail what's going on. Okay, it's red here, it's blue here, and green here. And we have put our primer here. We know what primer it is. We know the primer because the green piece of DNA, so this one, which is the adapter that we had before, has been designed by us. It is what we had put in the class before on the platform. So since we know the sequence of this one, it's very easy to design a primer that will hybridize here, and then the polymerase will start synthesizing this one. Okay. Now, how synthesis is working is as follows. Let's say that here is an A, so here it will put a T, but the T1 is linked to some fluorescent, and let's say that it emits red color. So here we have red, and here we will have red, and here we will have red, and here we will have red, and so on. So for the whole cluster we will have simultaneously the same color, which means that if this cluster that I have drawn here is on this spot of the cell in reality, so imagine that, okay, I drew it here like that, very big, because I want to have some space to make the drawing, but this one occupies a very, very tiny space on the original platform or the flow cell, so it must be somewhere here. So all these molecules simultaneously will give a reddish signal like that, and eventually what the machine will detect is a bright red color. And this synthesis step, since these nucleotides carry some fluorescent, it's a bit slowed down, and this guarantees, more or less at least, that there is a synchronization between them. So it is that all of them more or less simultaneously will give a red light, and this can be easily detected. So if adenine is red and guanine is blue and cytosine is green, and thymine is yellow, then what we will see is something like that. And so on. So the full platform here will be full of colors. Each color corresponds to a cluster of identical clones, since we have washed away the reverse complements. And the machine reads the colors and knows what sequence, what nucleotide we have there. So eventually, if we watch this process in time, so at time 0 we will have this image here, and then at time 1, exactly on this spot, we may see a green here. And then at time step 2, exactly on this spot, we may see a T here. Which means that for this specific fragment that we had on this cluster, we will have the sequence A, because this is red, and then C, and then T, and so on. And if we follow this order for all clusters, then what we will eventually manage to do is simultaneously get the sequence of millions of fragments. Okay, this process is described very nicely in a video made by Illumina that I will give you the link in the e-learn.